Assalamu alaikum. In this video, we are going to talk about vacutainers. So basically, hematology blood samples are sent to lab in either of these vacutainers, which are basically have different colors and there is purple top, green top, blue top, yellow top, gray top, and red top tubes. In each of these tubes, there is a separate chemical ingredient and that has different mode of action, and because of that each test requires a different kind of vacutainer tube. These tubes are called as vacutainers because they have vacuum inside it. So let's begin with the first tube, that is purple top tube. The chemical ingredient it has is basically EDTA. EDTA, its mode of action is that it acts as a chelating agent as they bind calcium for blocking the coagulation. Basically, this video is going to be easier for you if you have already the knowledge of blood hemostasis, or you have seen that video that I previously recorded and uploaded on blood hemostasis, because these tubes are interacting with the blood, and the blood has ability to clot, and it has a proper series of processes through which it clots, and calcium and these thrombin, fibrinogen, all these are involved in it. So these the chemicals that are present in each of these blood vacutainer tubes interact at different sites of those processes. So you should understand that hemostasis how hemostasis happens in those processes first. So purple top tube acts as a chelating agent as they bind to calcium, and when the calcium is blocked, the coagulation process will block. So in this tube your blood is not going to be clotted. So basically EDTA is calcium total chelate there, and that means that it is a strong chelator, and it inhibits calcium from activating all those coagulation factors and coagulation processes and platelet aggregation. So that was the mode of action. They Now, what are the indications for which we refer to the purple top tube? I will just name all of these, you can say, tests that require the each of these tubes, but I'm not going to get into the detail of these tests, because after this video we are going to start in the next series the biochemical chemistry panels, and there each test will be seen in detail. So the indications are CBC, ESR, blood smear preparations, direct comb test, hemoglobin electrophoresis, malarial parasite, ratic count, that is basically reticulocyte count, blood grouping, or G6PD test. Basically, it's a protein that is related to RBCs, and that leads to the hemolytic anemia if this protein is not available. Then cross matching of the blood, like major and minor crosses before blood transfusion. Then red cell folate levels. What are the folate level concentration in an animal or human blood? So for all of these indications, purple top tube is being preferred. Now there are some comments on this purple top tube. There are some advantages and disadvantages. The first disadvantage is that if there is an increased concentration of EDTA, like you can see this thing as that you have a purple top tube that requires 5 ml blood to be placed inside that bottle, but you place only 1 ml blood. So EDTA concentration in that in that purple top tube is basically for 5 ml blood, but you have placed 1 ml. So EDTA will act as a hypertonic solution for that RBC, that will cause RBC to shrink, and it will cause RBC lysis. So that is one consequence, that you have always to be prepared to or skilled enough to extract 5 ml blood from your animal, so that this problem don't happen. Next thing is that it lyses the RBCs of birds and reptiles. So if you have a bird patient or a reptile patient, you are not going to use this tube for CBC or any of these tests. It does not stain that is a now this third point is basically an advantage, that it does not stain the cells and does not interfere with blood cell morphology. So the cell preservation is optimal with this anticoagulant, and that it like this property basically makes it so good for blood smear preparation, that the EDTA is not going to interfere with your staining processes of your blood slides. So आपकी जो blood smear की slides हैं वो बहुत अच्छी बनेंगी, क्योंकि सेल की मॉर्फोलॉजी जो है वो डिस्टर्ब नहीं होगी इस केमिकल से. Next thing is that is also a consequence, that never use use purple top tube for the chemistry panels. Why? Because calcium is blocked and some chemistries need calcium to be evaluated. Then the EDTA interferes with a few tests like urine cortisol creatinine ratio test and troponin test. That's why in our indications we don't have any of these tests. Then EDTA falsely increases potassium and total iron binding capacity. So if you are also doing like blood chemistry, and you should know that the potassium will be falsely elevated. Similarly, EDTA decreases calcium, iron, potassium, Al, Kp, SDH. I am not going into the detail what are these enzymes because we will see that in biochemistry video lecture. But these factors are going to be falsely decreased. So you should memorize that these things, these changes will happen, and you should not rely upon that if your blood is being submitted in lab in purple top tube. Next tube is blue top tube, which has sodium citrate in it. Basically, sodium citrate is a chelating agent, but it is a gentle chelator than EDTA. So it will chelate, act as a chelating agent to the calcium, but it is a reversible calcium chelate. So clotting, if you have like blood sample in this a tube, and you want to reverse the clotting process, you can just add calcium, and the clotting process will begin. And that's why if you need these tubes, you have blood in these tubes and you want hemostasis testing, you can add back calcium in it and the clotting process will begin, and you can basically see the hemostasis testing, or like the process of hemostasis is going well or not. Like you can have all those values uh, with you. Now, indication for blue top tube is for coagulation profile. FDPs, basically this is fibrinogen degradation products. We have discussed their detail in previous hemostasis video. Then D dimers, APTT, NPT. Basically this is activated partial prothrombin time and prothrombin time. This is basically included in coagulation profile, and we will see their detail in coagulation testing video series. This is an indication for blue top tube. And one more comment on blue top tube is that it is not good for estimation of calcium, because first thing, you need exact 1:9 ratio of citrate with blood sample in this tube, and you should make this sure that you have 1:9 ratio. Otherwise your all results are going to be false, positive or negative, like false results you are going to have. Okay, next we have is green top tube. In green top tube, there is the chemical is heparin, and heparin basically acts as antithrombin 3, and it blocks thrombin and conversion of fibrinogen into fibrin. Basically, you can see that prothrombin is converted into thrombin, and thrombin converts fibrinogen into the fibrin. That's the normal process of clotting. But antithrombin, what does it do? That it acts on the thrombin, and it does not let function in a way that fibrinogen will be converted into fibrin. So basically, it stops the clotting process. So heparin, what does heparin do? That it also um, acts as antithrombin 3. It basically binds with antithrombin 3, heparin, and it will bind to antithrombin 3. When it will bind to antithrombin 3, it will basically accelerate or increase the activity of antithrombin 3. So the thrombin will be like blocked, and the fibrin is not going to be formed. So that's the mode of action of heparin. The indication for this top tube is if you want to have a renal function test, liver function test, anticoagulation test, if you want to check serum calcium, serum lipase, serum albumin levels, serum electrolyte and biochemistry, serum LDH, serum uric acid concentration, if you want to have hepatitis serology, lipid profile, if you want to have indirect comb test, or if you want to check the vitamin D3 level. So these all are the indications for the green top tube. Now there are few comments. First, you can use this for RBCs of birds and reptiles. In purple top tube, we have seen that we cannot use that top tube because purple top tube has EDTA, and that will lyse the RBCs of birds and reptiles. But you can use the green top tube. So if you are like a wild animal veterinarian, you can go for green top tube when it comes to dealing with reptiles. Then WBCs do not stain well because heparin binds to WBCs and platelet generally clump. So it is not recommended for DLC, differential leukocytic count, or hematologic testing because it will cause platelet and WBC clumps. So it is a bad idea if you want to have DLC or hematologic testing, and that's why we don't see any CBC in the indications of green top tube. Next slide, I'm going to show you a slide. Basically, you can see this, that when you have a blood sample with EDTA, you can see each individual cell. You can see the shape of cell, like this is a complete spherocyte type cell, but these cells have different like knob like shapes. You can see clear cut WBCs, RBCs, each and everything. But when the same blood sample is being stained with heparin, you can see you are unable to see any of the WBC. Then you can see how like dusty type stained RBCs are, and you are seeing these clumps of like strands. So heparin is a bad idea for CBC. So coming back, now there is a difference between heparin versus citrate. What's that? Heparin basically block the formation of thrombin. Citrate blocks the calcium by chelating. So overall, both are like acting or both are like blocking the hemostasis process. Both are trying to stop the clot formation, but they way or the method they are doing so are different. Heparin acts on thrombin and citrate blocks the calcium, and also EDTA also blocks the calcium. But difference between EDTA and citrate is, we have seen previously that citrate is a gentle chelator, EDTA is a strong chelator. So this was about green top tube. Next we have is gray top tube. Gray top tube has mainly sodium chloride, but sodium, its mode of action is that it is a weak anticoagulant, so it is used with potassium oxalate. You can see in this gray top tube you have this sodium fluoride and then potassium oxalate. Two things are added in these top tubes. So this, the mode of action is that it inhibits enolase and glycolysis. So it inhibits the glucose breakdown, and that's why it is used to measure the glucose and lactate. So the indication for gray top tube is used for glucose estimation in the blood of animal, and it is an enzyme inhibitor because it inhibits the enolase enzyme. So the only indication for gray top tube is glucose estimation, and the chemical it has is sodium fluoride and potassium oxalate. So the next we have is yellow top tube. Yellow top tube is called the chemical it has is a gel, that is a separating gel. Basically, yellow top tube is a serum tube. It is a clot activator, or you can say serum separating tubes. You also name yellow top tube as SST, serum separating tube. Why? Because the gel activates the clot formation, and when the clot will form, you are left with the serum. Up till now, the previous all the types of the vacutainers were the ones that used to inhibit the clot formation, but the yellow top tube is the one that will activate the clot formation. यानी कि वो clot को रोक रहे थे, क्योंकि वो लाइक उनमें क्लॉट के लिए चिलेटिंग एजेंट्स थे कि क्लॉट ना बने। येलो टॉप ट्यूब क्लॉट को बनवाती है ताकि जो क्लॉट है वो आपका बने और आपके पास सीरम बच जाए, बिकॉज हमें पता है कि ब्लड में जब भी क्लॉट बन जाता है तो जो चीज़ हमारे पास बच जाती है वो हमारे पास सीरम होता है, बिकॉज वी वॉन्ट टू check the things that are present in the serum. So some normally this yellow top tube has separating gel, but some serum separating tubes have silica crystals too. That's a clot activator too. So what will happen that you have blood in these tubes and you centrifuge the blood in this tube. So the gel is going to separate the clot from the coagulation factors from serum in that particular sample. So you have that clot being separated and all the coagulation factors are being like mixed in that clot, and you have your serum separated out in that sample. Now, what are the indications for yellow top tube are, like you check for vitamins, antibodies, serum ferritin, hormonal profile, tumor profile, cardiac profile, and CRP test. So basically CRP is C reactive protein test. So these are the indications for yellow top tubes. But there are different like school of thought also exists when it comes to hormonal profile, that it is said that some hormones will bind to the gel and falsely decrease their value. So you can also, like, it's preferable if you avoid yellow top tube. So although in its indications it's also mentioned that you are going to study in the indications of yellow top tube that it's for hormonal profile, but it's also being prohibited because some hormones will bind to that gel. So next, we have is red top tube. A red top tube is basically also a coagulation accelerator. It will allow the clot formation. It's same as a yellow top tube, but it will clot faster, so that serum will be extracted from it. Its indication are chemistry panels and blood studies. Now you can see if red top tube and yellow top tube both are serum separators, what is the need of red top tube, like how, in which condition we prefer red top tube? You should prefer red top tube in large animals, because large animals blood take a while to clot, and it is difficult to obtain serum from large animals like ruminants. Why? Because ruminant blood has higher concentration of fibrinogen, that is a protein that is involved in blood clotting, so it makes clotting process difficult, and that's why we don't prefer EDT or other like tubes or yellow top tubes. We prefer red top tube, that it has a coagulation accelerator that will allow the clot to form fast in such animals where clotting takes a while to happen. So uh, that was all about vacutainers. I hope you have fully understood the whole lecture. If you have any queries, you can comment below. I'll respond. Thanks for watching, and do subscribe.